Rates of suicide and major depression increase with altitude. Living at altitude involves chronic exposure to hyperbaric hypoxia, and healthy people living in Salt Lake City at 4,500 feet have lower blood oxygen levels versus those at sea level. Since serotonin synthesis requires molecular oxygen, our hypothesis was that hypoxia may reduce brain serotonin levels to worsen depression. We created a novel animal model to study altitude-related depression. Rats were housed for 2 weeks at sea level, 4,500 feet or 10,000 feet, and then tested for brain serotonin levels, for depression in the forced swim test, and for antidepressant efficacy of SSRIs. At altitude, forebrain serotonin decreased in female rats, but not males, and similarly, depression increased in female rats housed at altitude, but not males. SSRIs require adequate brain serotonin levels to function, and we found that at altitude, both sexes in our model did not respond to the SSRIs Prozac, Paxil, or Lexapro. The tricyclic antidepressant desipramine worked at altitude, but is not a clinical option given its severe side effects. Living at altitude may thus cause treatment-resistant depression, which is highly linked to suicide. We then attempted to find novel treatments for altitude-related depression. Creatine is a brain cellular bioenergetic compound, which is a marker for brain energetic health. A forebrain creatine deficit is noted in MDD patients and is also seen in people living at altitude and in rats at altitude in our model. Dietary creatine intake can improve brain energetics in healthy volunteers. Therefore, our strategy was to test whether dietary creatine could improve brain energetics and be antidepressant at altitude. Rats housed at 4,500 feet were tested for antidepressant efficacy of creatine and its lipophilic analog cyclocreatine, which has improved brain bioavailability versus creatine. We find that dietary creatine increased blood creatine levels in both male and female rats, but increased forebrain levels of both creatine and serotonin in females alone. Dietary creatine also improved depression in female rats at altitude, but not males. Cyclocreatine, however, improved depression in both males and females at altitude, despite improving brain serotonin levels only in females. To conclude, SSRIs form over 80% of antidepressants on the market, but may not work well in people living at altitude. Utah has the highest antidepressant prescription rates in the US, but despite this, depression rates remain high in Utah, especially in women. In our animal model, bioenergetic compounds can restore the brain deficit in serotonin seen at altitude and can also function as antidepressants at altitude. These data thus suggest that bioenergetic compounds may be better treatments for altitude-related depression versus SSRIs. This is the first study to ever test cyclocreatine in depression. Previous studies of dietary creatine show that brain energetics improve in healthy volunteers and that creatine can improve SSRI efficacy in MDD patients. Thank you.